Viruses, bacteria, and archaea. So we're getting into our first group of organisms. The bacteria and the archaea are actually living organisms that can survive in the environment. But before we get into that, we're going to talk about viruses. Viruses are a very unique group in the sense that they cannot live outside of another host cell, and we'll talk about that. So let's talk about just kind of how some basic characteristics of viruses. So we see viruses in many different organisms, plants and animals, and they require a host cell, which means they infect the cell of a plant or animal, and then they take over the machinery of that host cell and use it to reproduce and survive. So viruses are non-cellular, so they do not have organelles. They cannot survive in the environment. They can survive on surfaces for a short period of time, but they're going to die over time and become non-viable. They have DNA or RNA, so they're just small pieces of genetic material that is needed to reproduce and cause infection. We'll talk more about that. So we did not know that these were even around. We didn't understand that they existed, but the French chemist Louis Pasteur suggested that there was something smaller than bacteria that was causing disease, and he studied the disease rabies. So he was able to filter out bacteria and found there was still infectious material in solution, and that's where he determined it must be smaller than bacteria if they're able to pass through a filter that is small enough to filter out bacteria. So when you look at some common diseases you may have heard of, now this textbook PowerPoint was written before the coronavirus came out, so you're not going to see coronavirus on this list, but you definitely will see this in future editions of our textbook. But these are some common viruses you may have heard of: the AIDS, HIV, genital warts, which is what we give the HPV vaccine for now. We give genital and oral herpes, mumps, measles, chickenpox, common cold, the common sniffly cold that you get every year that's not coronavirus, that is just caused by a virus, so antibiotics do not work. Influenza, this is the respiratory illness where we get our flu shot. That's another illness that is similar to coronavirus. It's not the stomach bug, that's a different type of virus, but this is the type that infects the respiratory system and causes a lot of those problems that we see with the coronavirus. And SARS, so COVID-19 is a SARS type virus, so this is in that category of respiratory diseases. Skin diseases, so the typical warts you find on your skin that just don't want to go away, those are a virus that infects the skin. Fever blisters, that's oral herpes, so if you ever get breakouts around your mouth that kind of tingle and cause pain and have an open sore, that's oral herpes. And shingles is another version of chickenpox. Similar virus there. Gastroenteritis, this is the stomach bug. If you've ever caught an illness from some roommate or some family members that caused vomiting and diarrhea, that's typically the norovirus that causes that easily transmittable vomiting and diarrhea stomach bug that we call it. We incorrectly call it the flu because it's actually a stomach bug. It's a stomach virus. It's not influenza, which is a respiratory illness. So norovirus, diarrhea can be transmitted, for example, in airports and on airplanes, typically caused by a virus. Now, we do have vomiting and diarrhea caused by bacteria. That's known as food poisoning. And that usually occurs pretty quickly after eating an offensive food and resolves pretty quickly. Polio, rabies, and encephalitis have viral versions. And then other diseases that hopefully you've never had to deal with. Smallpox, we've eradicated. And I had my video yesterday talking about vaccines and viruses that we've been able to eliminate with vaccines. And cancer, some cancers are caused by a virus. For example, cervical cancer is caused by the HPV virus, the human papilloma virus. And that is why we give that vaccine now. Hepatitis is a virus that commonly infects the liver. Mononucleosis, maybe you've had mononucleosis before. That's the Epstein-Barr virus, causes widespread illness. Some of these other fevers are more in developing countries and tropical areas, yellow fever, dengue fever. Conjunctivitis is another name for pink eye. So that's a common virus that follows a cold and that causes pink eye. There is also a bacterial version of pink eye, so that can be treated with an antibiotic. And oftentimes antibiotic drops are effective against pink eye if the bacteria is the cause. And then another hepatitis, which is a virus that infects the liver. So when we look at viruses and how they're named, they can be named by what type of structure they have, and we'll talk about that, or the spike proteins that are present on the surface. So they are characterized, what makes them unique is what this means, is they are very, very small, 100 to 400 nanometers in diameter. That's very, very small. A nanometer is 1 times 10 to the negative 9, so that's really, really small. They have a core of genetic material, and it can be DNA or RNA in the core. And then they have, some of them are encoded by a special protein called a capsid, or some are not encoded by a capsid, and those are considered naked viruses. So we'll talk about that. So when you look at just the inner core of a virus, you have that DNA or RNA, that genetic material that allows it to reproduce and infect cells, and then it has different enzymes inside to help with metabolism and promoting survival. And then the outer covering is a protein called a capsid, and then an envelope that surrounds that capsid. So the envelope, like we said, is not found in all viruses, but there is a protein coating called a capsid around most viruses. So here's an example of some different virus structures. We see this one is called a bacteriophage. This is a virus that infects bacteria. So the capsid is this outer protein coat, and then you can see the DNA inside of there. And then there are some other special structures that help it infect and attach. So we have these pins here at the bottom, and then we have these tail fibers, and then you have the tail sheath. All of these are helpful for attaching and injecting this DNA into the host cell. The same thing we see here. We have different protein spikes on the surface, and there's that capsid outer covering that encloses the DNA inside. So lots of different structures to these viruses. This is really similar to the coronavirus. It has this envelope and it has these spike proteins on the surface. And the spike protein, this is what we have created an mRNA strand for, and that's what's in our vaccines, is the recipe for a spike protein to stimulate our immune system to make antibodies to that spike protein. But the entire virus is not in that vaccine, which is nice. It's only the spike protein that's enough to stimulate an antibody response and to get our immune system to recognize that virus. Should we catch it out in the community, it'll be rapidly dismantled and destroyed, and we're not going to get sick if we've had our COVID-19 vaccine. Here's another type, a structure of the tobacco mosaic virus. It's an RNA virus, so there's RNA inside of there, and it has this special kind of spiral helical shaped capsid. So you'll notice that every virus has this protein outer covering called a capsid, and it may have an envelope outside of that, or it may not. For example, this one does not. So this is a naked virus. This is an encapsulated virus because of that envelope that's surrounding the outside. So we call viruses obligate intracellular parasites. Obligate means it's obligated. It's required to live inside of the cells of the host that it infects. It cannot live outside of a living cell. So be sure that you highlight these terms here. It cannot live outside of a host cell. Very, very important to understand that. For example, the flu virus, when we're growing those to make vaccines, we have to use a chicken egg to grow that flu virus. We can't grow flu virus outside of cells. So it requires either a chicken egg or tissue cultures of some kind. So we need these tissues made up of living cells to infect with the virus, and then we can culture and grow them. To culture a virus means to grow it. So where did these viruses come from? We don't really know. There's a couple of different theories, hypotheses of where they came from. Excuse me. I'm not going to hold you accountable for these, but they don't really have an exact explanation for how viruses came to be. But they do very readily reproduce in our cells, and there's 5 steps. Number 1, first step is they attach. They attach to our cells via those spike proteins in the case of the coronavirus, and they gain entry into the through the mucous membrane into the cells, and then they take over. So they penetrate, they inject the virus into the cytoplasm of the host cell. So first they have to attach, then they have to penetrate, and then that virus, that DNA, will take over the machinery of the host cell, incorporate itself into the host cell's DNA, and then it will mature. By maturation, it will assemble new viruses inside of that host cell, and that will kill the host cell eventually. And then as that host cell breaks down, those new viruses that were created are released into the environment. So that's why it's so important that we stop a virus in its tracks and not let it get to this point where it can create new viruses inside of our host cells, because eventually that host cell is destroyed, and now we have that many more viruses. So we want to stop that reproduction as soon as we can, with either medications, antivirals, or with vaccination, so it doesn't start to infect cells to begin with. So there's 2 major life cycles of viruses. There's the lytic cycle, where we have viral reproduction and the host cell dies. It undergoes lysis, and lysis again means breaking apart, being destroyed, releasing those viral particles into the body. And then we have the lysogenic cycle, and this is where we still have the attachment and penetration and biosynthesis, but we don't destroy the cell and release that virus into the body yet. So it's kind of a latent period where we have the genome entering into the cell, but it's not destroying that cell yet. Later on, it can destroy the cell, and then it releases all those viral particles, and then the symptoms are present. So we see this in the HIV virus, the human immunodeficiency virus that causes AIDS. People can be infected with that virus for up to 10 years, and then it'll start causing the symptom of AIDS. So it's in the lysogenic cycle where the infection takes place, but it doesn't destroy the T cell, because this is a virus that the HIV virus infects T cells that are in charge of the immune system. So it infects the T cells, but doesn't destroy the T cells until about 10 years later when it enters that lytic cycle. So it spends 10 years in the lysogenic cycle and then goes into the lytic cycle, and that's where the T cells are destroyed, and that's where we see the immune system problems begin. So it's important that people be tested for HIV and get their treatment for HIV to keep it in the lysogenic cycle, and eventually it can be undetectable in the blood of people that are infected. They'll always remain HIV positive, but they can prevent from, or they can greatly reduce the chance of spreading and the chance of it going into the lytic cycle and causing the symptoms of AIDS by taking their antiviral medications. We've gotten really good at that medication, and HIV can be managed now today better than ever before. So this is just a little visual of the lytic and lysogenic cycle. This is a bacteriophage that is infecting a bacteria. So it's infecting, it's injecting. Well, first of all, it attaches. Then it penetrates and injects that DNA into the DNA of the bacterium. So here's the DNA of the bacterium. Now it's incorporating the viral DNA into the bacterial DNA, and it's making more and more of that virus inside of that bacterium. It matures and it's ready to be released. So it destroys the cell and releases those new copies of the virus. So again, this is the lytic cycle. And the lysogenic cycle is when the cell is just hanging out. It has the DNA inside of it, but it's not actively reproducing yet. So the flu virus is, again, similar to the coronavirus. We have this special virus that has this outer envelope, and it has these spike proteins. There's different types of spike proteins on the flu virus. There's the hemagglutinin spike and the neuraminidase spike. And we name the flu viruses based on the type of protein spike, the H and the N spike that it has on its surface. So you've heard of H1N1, but there are others, which is the swine flu. Then we have the H7N9, and we have the H5N1. These are very serious respiratory illnesses that are similar to the coronavirus, and we have to watch these very carefully and make sure we're not seeing outbreaks. Sometimes we've seen outbreaks of the avian flu, and people have had to destroy their entire crop. Their flock, I should say, not crop, an entire flock of chickens because of avian flu concerns. So the bird flu is another name for the avian flu. So it undergoes mutations, and sometimes that mutation can allow it to jump from only infecting birds to also infecting humans. So the spike proteins determine what cell these viruses can infect. So this is a human flu virus that has the spike proteins for infecting human cells. And if a worker, a farmer who has a chicken farm, for example, deals with a lot of birds that are infected with the bird flu, and those spike proteins are present just for infecting birds. But if that DNA is allowed to interact because of close contact with lots of flu virus, as well, bird flu virus, as well as a human flu, those DNA can interact, and form a new virus via mutations and genetic recombination to have a virus that can infect both humans and birds. And that's very serious because we don't have effective treatments against these avian flus, and that can cause death and, again, be something that can spread very rapidly through a population. So even though coronavirus is the big focus right now, the avian flu is always a threat. So these are emerging viruses, that we're aware of, but are not serious yet, but are getting serious. For example, when this textbook was written, they were talking about SARS, severe acute respiratory syndrome. This is a corona type virus. So the COVID-19 is a new version of a SARS virus, but it has already been identified as a potential threat, and it is a threat as we see now in our environment. So we have other ones as well that you may have heard of. Ebola was a scare, but now we have come up with a successful vaccine for that. And these are all able to mutate, and that's the biggest concern, is the mutation of these viruses. They're very good at mutating and becoming more transmissible and more likely to cause illness and death. So the more we let these viruses spread around our community and not get vaccinated, the more likely they are to mutate and become more serious and more deadly. So vaccination is extra important. So when we look at things like herpes, HIV, and cancer, these are the most common viruses that infect humans, and we cannot use antibiotics to treat those illnesses, so we have to use powerful antiviral agents. Prions, you may have heard of that. Excuse me, heard of that. These are small little protein molecules, very small particles, derivatives of viruses. Same things with viroids. Viroids are naked strands of RNA, so they don't survive very readily in the environment, but they do survive in living organisms and can be transmitted among them. So you've heard of mad cow disease. That is a problem, over in Europe. It caused the destruction of many, many cows there. And you've also heard of the prion that infects deer here in the state of Wisconsin, Minnesota, and Iowa, and those animals need to be tested. When anybody gets a deer, they need to look for that prion presence in the brain tissue to make sure that that animal is clear. So it's always important to register and check out your deer if you get one, looking for that prion disease. So we will stop here. That concludes the discussion of viruses. The next topic to pick up on are the bacteria, which are known as the prokaryotes.